Okay, there we go. So we stopped just short of the central dogma. And this is an example of how the Francis Crick has an extremely good opinion of himself and likes to make things dramatic, shall we say. But he is nonetheless pretty close to his dogma as biology gets. The central dogma was enunciated or articulated by Francis Crick in the 50s. And the idea here is that the fact that DNA can base pair within itself, that A can match to T, that G can match to C, and that every double helix is completely base paired all the way up. That's complementarity. The bases on one side are complementary to the ones on the other side. E-type complementary. Okay, it's the E-type complementary. And because the DNA is complementary, that allows for replication. So you can use one double helix to generate new molecules that are, again, complementary to the first one. So this one will have new strand on that side, and that one will be complementary there. That would be replication. It's complementarity that allows for transcription. Both RNA and DNA are nucleic acids, and therefore the base pairing between DNA and RNA can generate complementary copies. And finally, that there's a complementarity through adapters that's going to allow protein synthesis. That is, RNA is a nucleic acid, protein is a polypeptide, and in order to get from a nucleic acid to a polypeptide, there has to be some kind of an adapter to turn the information of a nucleic acid sequence into a protein sequence. And the fundamental aspect of the central dogma is one-way information flow. That you use DNA to make more DNA, then the information from the DNA goes into the RNA, and the information in the RNA goes into the protein. And for the most part, that is in fact true. There are some RNA viruses that don't bother with DNA. Reverse transcriptase can take RNA, make DNA out of it. But in terms of the basic housekeeping way that you and I work on an everyday basis, information flows one way. This is one reason why Lamarck was wrong, that you can't take the changes in the phenotype and push it back up into the genes. So let's take a little look at the complementarity during transcription, because this is something that I think can be disorienting for people. Here we have a molecule of DNA which the authors have thoughtfully given two colors to the strands. So there's a dark and a light purple one. And like all DNA molecules, it's got a 3' and a 5' end touching each other here, and same way at the other end. So that one of these strands is running 3' to 5', and the other strand is running 5' to 3'. Now, if an RNA polymerase molecule is bound to DNA and is making a new molecule of RNA, the RNA molecule itself will be synthesized from its 5' end towards its 3' end. But because of the anti-parallel character of nucleic acids, that means that the DNA is being read off 3' to 5'. It also means that the template strand that the polymerase is using as a source of its information, the template strand, has a sequence that's complementary to the mRNA. And that's why it's called the template strand. It's where one's high, it's low, and so forth. It's a template for it. The strand across the way that is not being used to make the RNA is called the coding strand, because it will have the same sequence as the mRNA. The mRNA is complementary to the template. The coding strand is complementary to the template. So the mRNA and the coding strand have the same sequence, except that it has U where the DNA would have T. I hope this helps you get oriented. I think the handedness of DNA molecules is one of the sources of confusion for people when they start to think about these things. Any questions about this picture before we head on? Okay. So, for this all to work, there has to be some kind of a code, some way to turn nucleic acid information into protein information. And in practical fact, the way it works in our reality is through a triplet code. Nucleic acids have 4 bases at a time. Proteins have 20 amino acids at a time. And so somehow or other you've got to find a way to map from 4 nucleotides to 20 amino acids. You can't have A stand for alanine and G stand for glycine. You'd run out of amino acids. Suppose you took them 2 at a time. Well, 4 in the first place, 4 in the second place. You'd have 16 possibilities. You wouldn't really have enough to do all the amino acids. But if you take 3 nucleotides at a time, you now have more than enough codons to specify the amino acids. And what that looks like is this. Here you have a strand of DNA. They're showing you the template strand here, the 3' to 5' one. It produces an mRNA reading like that from 5' to 3'. For each 3 of these nucleotide residues, there exists a transfer RNA, a tRNA, that is complementary to that triplet. So this triplet of nucleotides has a tRNA like this that happens to code for leucine, for the 1 amino acid of leucine. Here's another triplet that has a complementary tRNA for serine, and so on. So 9 nucleotides or 9 bases in the mRNA produces 3 amino acids in the protein. So 3 bases at a time will be a codon of nucleic acids, and a codon can specify 1 amino acid. This is not in your textbook, but I thought this was an interesting way to represent the triplet code. They tend to always do it as that square box, and there are drawbacks to that. One of the things I think comes out of this particular representation is it really brings home the extent to which the genetic code is a very sensible code. It really takes advantage of the fact that it's a triplet code. So let's say we want to code for an alanine. The tRNAs for an alanine the codons for alanine are going to be a G, C, and then anything in the 4th position. So one of the things that this is showing you is that alanine has at least 4 codons, but that it's only the first 2 positions that matter to figure out that that's alanine. Here's serine with a lot of codons. Here's isoleucine with 3 codons. Here's methionine with 1, which is going to turn out to be quite interesting. We'll look at that later on. Proline has a lot. Tryptophan has 1. Cysteine has 2. So there are different numbers of codons depending on how abundant an amino acid is, pretty much. And so that's one of the flexibilities of it. And the way it's organized means that a mistake in the 3rd place is less likely to cause harm at that position. It'll be a synonymous codon. That they both code for the same amino acid. The protein would never know there was a mutation. Is that clear? Any questions about that before we move on? The triplet code is even more beautiful than that, though. And it takes a little bit of explaining to show you what's going on in this picture. Part of it is, as I said, that there are many codons for some of the amino acids. So you can see this black line around leucine and so on to show that there are lots of ways of saying leucine. But what the lines are showing you is what happens if you mutate that codon. And what you see if you look here is that if you mutate leucine, you either stay the same because there are a lot of codons for it, or you turn into some other hydrophobic amino acids. If you're an asparagine and you mutate, you tend to stay in the same general neighborhood of polar amino acids. And so the particular pattern of code ends up being one that makes it especially robust. It makes it especially difficult for a mutation to alter the protein in a functional way. It didn't have to be that way. We're so familiar with the genetic code, we think of it as, well, of course that's the way it is. It didn't have to be that way. If you take a random drawing of all the 64 codons and match them up one at a time to a randomly drawn amino acid, only 1 in a million times will you come up with a genetic code that's as beautiful, as functional, as robust as the one we actually enjoy. This is showing one of these alternate genetic codes over here that came out through one of these Monte Carlo runs. But for the most part, you're not at something as good as what we've got. I have an article about this that I'll try to put on Icon for anybody who wants to read more about it. I think it's really quite beautiful property of the code. Any other questions about that before on? Okay. Now, it even gets more complicated than that. So all protein is coded in DNA. Any protein in your body goes back somewhere to a DNA molecule. But in us, most of the DNA does not code for protein. So if we were to look here at a picture of the total composition of the DNA in our bodies, 3/4 of it is never transcribed. It's replicated every time the cell divides, but it's not transcribed. About 1/4 of it is transcribed, but of that quarter, this little sliver here, about 2% of the total, actually gets translated into protein. There are a huge number of RNA molecules that are transcribed and then broken down inside the nucleus and never get out. Now, part of that is the processing of messenger RNAs. The initial mRNAs start out much bigger than the final product. But it's also that there are some RNAs that have no product at all in the cytoplasm, and we're still trying to figure out exactly what that means. It's one of the little spoken of mysteries that's been around for about 30 years. So what does this look like in practical terms if I want to make a protein? Let's look at a gene here that might be 250 base pairs, good sized. It gets transcribed into an mRNA that at its initial size is also 250 nucleotides. But a process can happen called splicing, in which bits and pieces of that RNA are taken out and it's put back together into a shorter thing. And we'll talk about that in more detail when we get into transcription later in the semester. So an RNA that started off as 250,000 nucleotides might end up as a mere 6,000 by the time it's a fully processed and spliced messenger RNA. Within that messenger RNA, only part of it may have codons. This is showing you, this would be the 5' end and the 3' end. This is showing you a good-sized 3' tail. There are messenger RNAs where the untranscribed 3' end is 3 or 4 times as long as the part that makes protein. So the nucleic acids, the RNA, does more than just make protein. There's information in it in different places. So once we have these 444 bases, they are going to give us about 1,500 codons. That many codons will give the same number of amino acids. So there'll be 3 times as many bases as codons, but 1 codon for each amino acid. I had that little asterisk about in complex organisms. That smallest eukaryote that we talked about back in the first chapter has a genome that is extremely spare and has apparently almost no garbage DNA and very few, very small introns. So it's partly a matter of the life history of the organism, how much of this mysterious RNA you're going to have around, or mysterious DNA. Yep. Okay, whoops, let's go back. Okay. All right, so there's the reading out the code. Now, one thing that is not really emphasized so often about the central dogma is that it's something that is called recursive. That is, you need it to make it. And if we look at just the information flow with the black arrows here, the information from DNA goes into DNA, the information from DNA goes into RNA, the information from RNA goes into protein. But for replication, to make more DNA, you need RNA and you need protein. To make RNA, you need RNA and you need protein. To make protein, you need proteins and RNA. So these processes are tangled up in each other. Each of these information flows uses several of the components. It's part of what has been so challenging in trying to figure out how life could have gotten started. And I think we'll have some time later in the semester to show how we might be able to simplify this to something that is a little more interpretable. Okay. Now the DNA in our cells is not just loose and like a big pile of spaghetti. It's packed up into chromosomes, so that we, for instance, have 46 chromosomes in each cell. And those chromosomes are not all identical. They all have a distinct shape, a distinct length, They'll all have a distinct pattern on them of activity. And this is showing you a diagrammatic map of the chromosome 7. It happens to be the chromosome that has the cystic fibrosis gene on it. And this is a diagrammatic way of showing what can happen after certain kinds of staining that will highlight the active or the inactive parts of the chromosome. And since the chromosomes have distinctive areas that are active, that gives each one of them a particular pattern. Cytogenetics is looking at genetics at the level of chromosomes. What chromosomes does this organism have? What chromosomes does this cell have? Here in this one you can see there's been a translocation where a little bit of this chromosome has ended up onto the end of that chromosome. Here's another translocation where a little bit of that chromosome ended up over there. Here's a really messed up genotype. And if I remember correctly, this figure may be the one that is showing how human and gorilla chromosomes compare. There's been kind of a patchwork of trading off bits and pieces of the chromosomes where you can still find most of the information, but they're not organized quite the same way. But we don't have to stop at the level of the chromosome. It's handy that we can detect chromosomes and tell one chromosome from another, and sometimes it's very important. That's how what amniocentesis is done for. But we can go down much finer detail than that. We can sequence DNA. We can know the exact order of the nucleotides in the DNA. Now, you've grown up with hearing about DNA being sequenced, so it probably doesn't sound amazing to you anymore. But in each of your cells, there's this much DNA in actual length. There's a meter of DNA in each of your cells. And there's 1 billion bases, base pairs, in each of those nuclei. And yet we can sequence that molecule. I mean, think of how skinny that thing is if I'm hanging out like that. And yet we can sequence that, and the total genome of the humans has been sequenced, as well as a number of other organisms. So, what do we need to do to sequence DNA? Well, you need to have a source, and to the source you're going to add some way of making more DNA, but in a very selective way. We're going to add a primer. So the primer will be a short piece of DNA or RNA, depending on the method, that will start the process of sequencing. That this is where I will start. The sequence of the primer determines what gets sequenced after that. You have to add molecules of DNA polymerase, and so we can now make pure forms of this enzyme that can be used essentially as reagents. Sorry. We need deoxynucleotide triphosphates, so DNTPs would be the deoxy form of adenosine triphosphate and so forth. And there are going to be a few fluorescently labeled dideoxynucleotide triphosphates. And we're going to see why we would do that. So when you mix all that stuff together, the primer will bind to the DNA, and the primer will provide a location at which the polymerase can start adding more molecules. And so initially, all that the DNA polymerase will do is match up a complementary base to each one that's there. There's an A, so to put a T in. There's a G, so to put a C in, and so on. So it's just reading on down, adding a base that's complementary to the existing base. That by itself would not give us a sequence. But you remember I said we have some dideoxynucleotide triphosphates in there. And this is the structure of a dideoxynucleotide triphosphate. So it has a base, like the ordinary ones. It has the sugar ring. It has the three phosphate groups. But instead of being just a deoxy here at the 2 position, as a deoxynucleotide triphosphate would be, it's deoxy here and at the 3 position. Now, I've been talking about nucleic acids from 5' to 3', so this thing doesn't have an alcohol on the 3 position. So as the polymerase is going along, once in a while, it'll accidentally incorporate a dideoxynucleotide triphosphate instead of the regular kind. But because it's got no alcohol at the 3 position, that stops the chain from elongating. That ends the process of elongation. There's no place to add the next one, so it stops right there. If you put into that mixture fluorescent dideoxynucleotides, you can have a different color depending on what nucleotide was put in. And so here it incorporated a T at that spot that stopped the polymerization, and the T's have been labeled with red. And here it incorporated a C, and they've been labeled with blue, and so on. So you get a population, a mixture of from very short to very long, pretty much randomly, although there are some exceptions to that. And there'll be one for each length along the way with a colored nucleotide at that position that represents what was in the original template. And so if you do that, you end up with something that looks like this. You see many little bands here, and each band is a band of DNA that stopped being polymerized at that spot because here it incorporated a T, for instance, or here it incorporated a G, whatever that color coding was. So each of these, it represents a DNA that's one longer than the one before. So this is one longer than that, that's one longer than that, and so forth. And you can see then that if you just go up here, counting, reading it off, green, green, yellow, yellow, red, yellow, yellow, and so forth, you can literally read off the sequence of the DNA. I can't begin to tell you how much easier this is than what used to be done. So you can see from this that you can sequence many things at once. So on this particular gel, you've got 8 or 10 that are being done at once. Some of the multiplexers now run, 20 to 30 at a time, Susan? Yeah. So quite amazingly powerful. DNA sequencing has gotten so good that it is now a tool of ecologists, so that if people want to go out into the ocean and figure out what's out there, instead of trying to isolate them and grow them anymore, they sequence everything. They just do shotgun sequencing. And in doing this, they've discovered that there are hundreds and thousands of species of prokaryotes that no one knew were there. So it's a fascinating time to be following these things. Now, the process I've described so far can't work on DNA this long. And so you need to have it broken up to some extent, but by using primers at different places, you can then have overlapping pieces of the original, and by noticing where they overlap, so this overlaps here and that overlaps there, you can reconstruct what the original sequence was. Many times when they're doing this, they may sequence, 6 to 8 times over each of those stretches to make sure they've got the exact sequence. We've gotten to be very powerful with DNA in the lab. You think again about a piece of DNA this long. We have enzymes that can identify specific locations on that called restrictions enzymes. So they recognize sequences anywhere from 4 to 8 nucleotides long. Do you know any longer than 8, Susan? Yeah, okay. And so if you have a sequence like this, for instance, it could end up being cut in a pattern like this, in which case we say it has sticky ends, because obviously that could re-anneal to that. There are also some times when it gets cut straight off, and these are blunt ends because they've cut right at the same place on both sides. Something with sticky ends will tend to stick back to itself. Something with blunt ends, you could probably mix with other things and get other things to come in. And because we can do this kind of thing, we can now recombine DNA to make the molecules we want out of it. So for example, if we start off with a circular piece of DNA called a plasmid, this is like a mini chromosome in a bacterium, and a piece of DNA for some gene that we want to make more of, an antibody, whatever, we can cut into that plasmid with restriction enzymes that identify those sites on both, and then put just that piece into the plasmid, ligate it with appropriate enzymes, and end up with a new piece of DNA, which is now called recombinant DNA, because we've recombined the original plasmid and something else. This is just showing you how you can then analyze things like that. Restriction enzymes would have a specific pattern of cutting in each of those, and by looking at their sizes on a gel, you could assure yourself that you do or don't have the right pattern in the plasmid that you've made. A lot of this has gotten easier because a number of people have designed things called cloning vectors. And the general logic of a cloning vector works something like this. We're representing here a circular piece of DNA. This happens to be PUC-18, University of California, San Diego, I think. One of the University of California universities made a whole bunch of these cloning vectors in the early days, and many people are still using them. PUC-18 is one of the good ones. And it came as a plasmid that existed in bacteria just because it was there. Added into it was put a resistance marker. So this is a gene to make ampicillin. And ampicillin would normally kill the organism, but now if they have this plasmid, they'll live because the ampicillin will protect them. That's just so you can tell where the plasmid is. The interesting part will be up in here. So there's a gene called LAC-Z. LAC-Z is an enzyme that's part of the LAC operon, and we'll talk more about it later, but it allows you to see whether anything's been added to it. So you can see here that there's a place inside of the LAC-Z where there's restriction sites. And so you can tuck your gene of interest inside that. And as a consequence, you can distinguish between the plasmids that do have an insert and the plasmids that don't. And so the end product might be something like this, where you can see plates that have colonies, and those colonies can then be analyzed in more detail. One of the more powerful techniques of modern molecular biology is something called polymerase chain reaction, or PCR. And the basic logic behind PCR is that you start off with some target sequence, and you have two primers. You have a primer going in one direction and then another primer in the opposite direction. You run the polymerase through that, and at the end of that cycle, you're going to have now two molecules, with a little bit of sticky ends there out, but that'll is not important because we're only interested in the dark blue part. If we then again add primers and again polymerize, now we have four molecules that are identical to the original, except for the sticky end. Do that again, now we'll have 8, and so on. So through PCR, each time we cycle through the polymerase reaction, we double the number of molecules. The way we can stop the polymerase chain reaction in between rounds is that we can heat this up to a high temperature and melt the double strands apart, and now they're exposed to the primers, and they can pick up primers and be targets for another round of polymerization. That's only possible because there's an enzyme isolated from a thermophilic bacterium, one of these things that lives in boiling water in Yellowstone. And the original PCR was done with painstakingly purified ordinary DNA polymerase, which was killed at the end of each round, so they had to add more enzyme at the end of each cycle. PCR is an exponential process. It doubles at each fold. And exponential processes are often dramatic. So population growth in the early stages is often exponential. The basis for nuclear explosions is exponentially sending off 2 neutrons for each neutron you get in, and so on. If you do 10 cycles of PCR, you have about 1,000 copies of what you started with. If you do 20 cycles, you have about 1 million copies. So it really can amplify to an extraordinary degree. We can use PCR in a really huge variety of ways. My husband is so fond of it. He says, if you haven't asked yourself how the experiment would be easier with PCR, you should stop and think. So this is just a sampling of some of the things that we can do with it. One is that we can create mutations. We can create the particular mutation that we want through PCR. So you can start off with your original plasmid. If you put in primers that are containing the information you want it to have instead of the original one, after you do many rounds of PCR, most of the copies are going to be the sequence you want it to be, and you can prove that it is by sequencing it. So this is a fairly powerful way to do very specific mutations. It can detect pathogens at levels that no other technique can. If you can amplify something 1 million or 500 million, obviously it's going to be more sensitive than something that just uses antibodies. One of the ones that you're probably all familiar with is the fact that it's used for criminal identification. The one thing we cannot change about ourselves is our DNA, and it's almost impossible not to leave DNA behind wherever you go. And so PCR has made it possible to take these tiny remnants of hair flakes and skin and stuff and identify individuals based on PCR of that DNA. I mentioned that we each have like 1 billion, nucleotides. You can't really look at 1 billion at once. And so a more common way of looking at someone's DNA is to take advantage of something called restriction length fragment polymorphism, or RFLP. And what that means is if you think about restriction fragments, so here's a piece of DNA, and in this person there are 3 sites where that restriction enzyme would cut. And in this one there are 2 sites. Then when you cut that with the restriction endonuclease, you'll end up with 2 pieces of DNA here and 1 piece of DNA here. And when you run that on a gel, these 2 smaller pieces will run ahead of the 1 longer piece there. This is polymorphism because there's differences between people, and that's what geneticists call that. It's fragment length polymorphism because these are the fragments that have been made through restriction digests. And what this means is that if you look at a number of these particular locations, individuals can be sorted out from each other by finding out which RFLP you happen to have. It's behind some of the things that are used for paternity testing, for instance, and so on. If you do enough different RFLPs, the confidence with which you can say it's you and not you can be very high indeed. It can be used to diagnose genetic conditions, so that's very handy. The efficiency with which it identifies individuals to some extent depends on the size of the population that you're looking at. That is, if you have a small group of people that don't vary very much, the odds of having the same RFLP are higher than if you have a large population with a lot of RFLPs. So that has to be taken into account. And this is just showing you an example of one of these, looking at a crime scene, for instance. Here's the victim with the victim's pattern. Here's the blood stain that was left at the location, clearly not from the victim. And here are 3 different people who might have been there, and obviously only one of those 3 matches in detail. There have been places, for instance, in England, where they're concerned about a crime and they make everybody in the village give a blood sample so they can identify RFLPs. That isn't done in America, and I think that it would be an interesting question if anybody wants to think about it, of, to what extent does the right to privacy to your own DNA have to be weighed against, for instance, a serial killer who may kill someone again. It's something worth thinking about. In the last few years, it's become possible to use organisms that are very well characterized. All of these organisms on this page are organisms who have been completely sequenced, so we know all of their genes, and that can be a very powerful tool to take advantage of. But they're good for different purposes. Each of these has, in a miniature version, the fundamental properties of some group, but it has it in a way that makes it easier to live with it or grow it or to do experiments and so on. So they're going to differ in size and they're going to differ in complexity. The smaller, the less complex. For many kinds of experiments, an E. coli, a bacterium, is a perfectly adequate model, depending on something that you're just going to be looking at, either a metabolic trait or something like that. And so you can do quite a lot of analysis at that level. But it is fundamentally a prokaryote. It is unicellular. So you start to have other questions that don't really work in E. coli. The next up on this scale here would probably be this cell. This is a yeast cell called Saccharomyces cerevisiae, or beer yeast. It's a budding yeast. You can see little bud scars there. They are true eukaryotic cells. They have true nuclear membranes. They have true Golgi. They have true ER and so on. But they're single cells, and they have a fairly simple lifestyle. They do have a little bit of sex, but not very complicated. Nothing compared to my thesis organism. And so if you want to ask questions at the cellular level, Saccharomyces is a good model. It's also a place that can be used as a way to probe for interacting proteins and so on. But there are other levels of questions that really can't be answered there. If you want a multicellular animal, one of the good models is C. elegans, which stands for Caenorhabditis elegans. And this is a worm. It is transparent, which is handy. You can watch it develop. This is one of those multicellular animals that is smaller than the largest prokaryote. So it's a small worm. And it can have a complete life history in a couple of weeks. So you can do your experiments in a very prompt way. But it does development. It has a surprisingly large number of the genes that we have. It may have one copy where we have 4 or 6, but it still maps out for a surprisingly large fraction of things. Oops, sorry. Obviously another model organism would be fruit flies, but you're all already familiar with them. This is something called Arabidopsis thaliana, which is a plant. And it is not quite that small, but it's quite small. But it has a complete set basically of everything. It's got flowers, it's got leaves, it has stems, and so on. And so it again is a very powerful method for looking at development or for interactions within different systems. Any questions before I move on? Okay. So what does it take to make an organism? How many genes does it take to make people or things? This is just a sampling of some of the different organisms. We have just shy of 3 billion base pairs. This is roughly 2,900 megabases, so a fairly good-sized genome. We have on the order of about 30,000 genes. Remember I said that only about 2% of that DNA ends up as making protein. And what this means is that there's about 1 gene for every 100,000 bases. It's sort of like Nebraska. It's a long way between gas stations. We happen to pack it into 46 chromosomes. If you look at a rat, it's really not very different. Any mammal is going to be roughly as complicated as a human being, bigger or smaller, but roughly as complicated biochemically, roughly as complicated at a cellular level. Not as good at conversation, but roughly the same. It starts to change as we get to things that are either less complicated or shorter lived. So if we look here at fruit flies, that's quite a lot less DNA, 180 megabases. Fewer genes, less than half probably, something like that. And now there's a gene in roughly every 10,000 bases. So we're now getting close to, rural Iowa for gas stations. Only 8 chromosomes here. Arabidopsis, that little teeny tiny plant that I showed you, has around the same, a bit less DNA. Actually has more genes. They recently sequenced one of the trees. It was either aspen or, do you remember which particular one? It was one of the ones that they're thinking of for biofuels. And it turns out that this tree has 30% more genes than we do. It's more complicated to be a tree than to be a human being, apparently. But then again, I can't photosynthesize, so who am I to talk? But in the Thaliana, again, they're fairly close together. Here's that little worm we were talking about. It has, about the same as the fruit fly, less than the plant. Again, same general density of genes. Here's the yeast plant. Less again, quite a bit less DNA. The genes are somewhat closer together, but that's because there aren't nearly as many genes. And we're getting down in here to about the simplest level we're going to look at for a eukaryote. They're actually sequencing that little thingy I showed you, the Austriococcus, that's less than a micron across, so we'll know more about that. But there's a big jump when you start to go down into the prokaryotes. So here's E. coli with just over about 5 million bases, half again as many genes, 1 every 1,400 bases. So you can see that the overall complexity of the organism doesn't, in general, relate to how many genes it takes. But there's also this odd relationship with the density of genes, that the more complicated you get, the more extra DNA is in there. We don't completely understand what's going on there. I mentioned that we have been mapping genomes. The human genome has been mapped now since 98, 2000? Sorry, what? Yeah, somewhere around 98, I think. And since that time, more and more genomes are being rolled off all the time. When you look at a human genome, some of the aspects that make it interesting or tricky are things like the repetitive DNA. Trying to do the human genome, they had to work out techniques for how to keep sequencing when you have the same sequence 20 times in a row. That wasn't a lot of fun. You also have the case that you have some of it that will be transcribed, but not translated. And then some of it will actually have genes. Some of the DNA will be there. And you can figure out what's a gene and what's not a gene in a couple of ways. One is whether it has an open reading frame. So an open reading frame is a stretch of DNA that has a start signal here and a stop signal here and a bit of a signal up there for the ribosome and so on. That it's one in which you have the potential to make a molecule. You don't know that you are, but it at least has the potential. You can also do it through expressed sequence tags. So these are cases where they've sequenced bits of DNA complementary to mRNAs. And since they're complementary to mRNAs, you know they got expressed. You know they at least made a messenger RNA. Maybe they never got to making protein, but at least they made RNA. And the third way is by seeing homology. That if you see a region of DNA that is highly homologous to something else, you can make a decent guess at it. And what you can see here is an example of what these genome maps end up looking like. So you can color code the genes for the different broad categories of function. You can see that in this particular place there's a fairly dense number of genes. They may go either direction, so you've got some going that way and some going that way and so on. So the final output from these is a presentation that is very dense in information and takes a lot of looking and thinking to start making it make sense in any complete sense. If we do that kind of thing and we look at, for instance, human beings, no, this is E. coli, I'm sorry. And here's E. coli. And E. coli has been under cultivation for over a century. It's a single cell, it's a prokaryote, it's a parasite, for God's sake. And so you'd think we could make sense out of it. And a lot of it makes sense. Here you can see that about 20% of the DNA is going to metabolism, just daily housekeeping, eating, cooking, and making stuff. About 4% to regulation. That kind of makes sense. About 8% to all the macromolecular processes, making DNA, RNA, and protein. Here's about 5% for structure. That makes sense. E. coli is a very boring cell. I don't want to offend any microbiologists in the audience. But you have to admit, there are a lot more exciting prokaryotes. Yes? Yeah, okay. 10% on transport. 10% of the genes are involved with transport. And then there's 38% that we don't know what it's doing. So it'll be fascinating finding out what's living in that great unknown over there. The smallest cells that we found so far have about 250 to 300 genes. That seems to be the smallest thing you can get away with and still be a cell. All this technology has then made it possible to make transgenic organisms. Transgenic organisms are one in which we have put genes into them for whatever reason. In some cases, it's for research, where we want to know if this gene is turned off or turned on, what happens. And there's a wide variety now of mice that are engineered to have a gene turned on or off. Some transgenic organisms are food organisms, and this is an interesting one over here. This is called golden rice, and by the kinds of manipulations we've been talking about up to this point, they inserted the genes, plural, for the pathway that makes vitamin A, and inserted them into rice so that this rice now makes vitamin A. And this rice makes enough vitamin A that it can prevent deficiencies of it in people who eat it. There are medicines that are being made transgenically. One of the more recent ones is transgenic chickens that have the genes inserted in them for some of the proteins that are used to fight cancer. And they were very smart. They put it in front of the area where you make egg proteins. So these genes put all their medicine into the egg. So you don't have to kill the chicken to get it. You just crack the egg open and purify it out of the egg white. And there's still obviously some ethical questions that come from this. One last reminder, if you'd like to do honors designation, please do come and talk to me. I'll be here to talk about any aspects briefly, and then I'll be up in my office after that's taken care of.
Okay, browser access. All right, thanks. Okay. Actually, I didn't know where. Well, I was actually thinking I was a collector, and then I was wondering if I can have a book. But I remember I heard an article about so maybe.
No, I think they did that. Oh, I think they did that growing inside of a square container.
Oh, that's boring.
But they do have growing green fish. They have transgenic zebrafish.
What's up, man? I gotta grab one. I'm gonna put them all.
So maybe research.
Hey, we gotta, dude, I'm trying to home. We gotta.
Hey, Ben.
I've been good. I'm good. Yeah. Glad to get back.
Go to the honors programs.
Do you have any other ideas?
Oh, there's. What are your interests?
I did. Well, the only thing that I just think of this, it was just so interesting that.
That's a great place. If there are any diseases in your family that you know about, there's a novel about diseases.
Yeah.
Want to write science fiction? I have a science fiction character.
Well, I'll look up the